for introducing me, and thank you to Gustavo for inviting me to give the talk today. So I will give a little bit of update on how ENCODE is trying to annotate non-coding variants and annotate the human genome, human and mouse genomes. So if you're not familiar with the ENCODE Consortium, it is an international effort with the goals of, number one, cataloging all functional elements in the human and mouse genomes. These would include transcriptional regulatory functional elements and also post-transcriptional regulatory elements. And these are many types of assays that the ENCODE Consortium is trying to produce, including DNA sequencing, attack-seq assays, which probe chromatin openness, chromatin immunoprecipitation, followed by sequencing experiments on either histone modifications or transcription factors, and DNA methylation, either whole genome bisulfite sequencing or targeted regions RBS sequencing. And also, in addition to try to annotate these functional elements, we are trying to, one main goals of ENCODE is to make the data as well as the resulting analysis results available as resources for research communities that study human and mouse, and also trying to use the knowledge to understand human diseases. So if you go to the ENCODE portal, which is encodeproject.org, you can have an overview as to the kind of data sets that have been produced by the consortium. So right now, in aggregate, roughly 12,000 data sets are being produced. As a disclaimer, that includes roughly 3,000 data sets that were produced by the Roadmap Epigenomics project, an independent project that has concluded recently, but the data are being ported into the ENCODE portal. So you can see these are the assay types. I mentioned some of them: ChIP-seq, DNA-seq, RNA-seq, and small shRNA knockdown of an RNA binding protein followed by RNA-seq. ECLIP is an assay for probing the locations of RNA binding proteins. As you can read, I can't quite see from this angle. And then the rows here indicate the cell types. So we are concluding phase 3 of the ENCODE project. And in phase 2, we had a lot of data sets on immortalized cell lines. It has been a major expansion in ENCODE 3 to generate more data on tissues and primary cells, and also whole organs. So you can see that these additional data sets go beyond the immortalized cell lines. So just a little bit about how the consortium is set up. So we have quite a few data producing groups that produce a lot of the data, which I showed you. And then there is a data coordination center headed by Mike Cherry at Stanford University. And then there is a data analysis center, which Laura mentioned. I'm heading that. And then there are quite a few computational analysis groups that we interact with in the consortium for analyzing the data, generating the analysis results. And for example, like these 3 indicate example analysis results, and then we collectively call these the encyclopedia. That's like the E in the acronym ENCODE. So if we talk about ENCODE Encyclopedia, is how do we go from these tens of thousands of genome-wide data sets, including ENCODE and the Roadmap consortia, And how do we go from here to integrate the different experiments and assays and find functional annotations, and how do we present these functional annotations in a user-friendly format so that we can help the community in doing their research, doing their clinical work, and so on. So in an effort to put together this encyclopedia, we have come up with these kind of tiered structures. So in the ground level would include results that are generated by a whole set of uniform processing pipelines. So for each data type generated by ENCODE, we have make a lot of effort to come up with uniform processing pipelines so that we can run through these data in a principled manner. For example, we have a DNA-seq has a pipeline, and histone mark ChIP-seq has its own pipeline. TF ChIP-seq has a pipeline. RNA-seq has a pipeline. So all these data sets are being run through these pipelines. And then the first layer of results are being incorporated into the ground level of the encyclopedia, because we believe that users will be much more easily being able to use these kind of interpretation rather than raw reads. Obviously, there are users interested in the raw reads. They can do so and directly download the data. But for people who don't want to process these data, they will be more interested in this ground level interpretation. So in the middle level, so when you go levels higher and higher, you are more and more integrating across different data types between annotation and raw data. So in the middle level, we're trying to interpret the data furthermore, trying to come up with regions in the genome that are like promoters, that are like enhancers, that are like insulators, like silencers, and then when you go further up, we start to have chromatin states. As you might be familiar with, we have ChromeHMM and Segway algorithms for calling chromatin states. We start to annotate genetic variants, whether or not they are going to impact functional elements, and allele-specific events, so allele-specific binding events or expression events, and also the target genes of enhancers that are in the middle level, enhancer-like regions, whether or not these regions target which other genes. So going further up, we have more and more interpretation of the data. So if you go to, again, encodeproject.org, you will see that in the ground level annotation, we incorporate all these results in these categories, and then you can go and browse the sets of data and download them or browse them using, say, genome browsers, the UCSC genome browsers. And for middle level, we also provide methods and then also visualize these data sets. And top level, we have chromatin states. For example, this is a figure from Manolis Callas' lab on epi logos for a whole set of chromatin states across many cell types. And then methods such as RegulomDB, HaproReg, and then FunSeq for interpreting both somatic, so FunSeq is somatic, or germline variants, what kind of impact they may have on regulatory potential, for example, here. So just to focus today's discussion, I want to give you a bit of update on how we define enhancer-like regions and also the target genes of these enhancer-like regions, so that it's quite relevant to the enhancer-like regions definition is quite relevant to the dream challenge that you will hear from Ansho right after me. It's not the same, but in the same vein of goal. So I thought this would be a good area to talk about. So this is actually a really great meeting. So for me to come here and it's quite a good eye-opening experience to see all these very sophisticated methods for doing all kinds of predictions of many different kinds of biological problems. So it would be good to incorporate quite a few of you guys' sophisticated computational methods into, for example, building the ENCODE Encyclopedia. So you may feel like the method I'm going to tell you about is actually really, really simple. But nonetheless, it actually, we had a lot of discussions about what kind of method we should use. And in order to predict enhancers genome-wide, because ENCODE has hundreds of cell types, and we want a method that's unsupervised and very simple so that we can apply the method to both human and mouse species and also across hundreds of cell types. So we want a very simple method. And we have also tested quite a few single feature just ranking schemes, and we found that among the many epigenetic signals that we have whole genome data sets for, DNase, which is, I mentioned earlier, for open chromatin signal, and the ChIP-seq of H3K27 acetyl, this is a histone modification. These two data sets, these two features are the most predictive of active enhancers. So for quite a few cell types in ENCODE, we have both DNase and K27AC data available in the same cell type. And for additional cell types, we have one of these two data types available. So we came up with a method that combined these two data types and then to extrapolate to as many cell and tissue types as possible. So even for a very simple method, as you heard from earlier talks, still like what kind of parameters to use, exactly how do you formulate the method, still require a lot of care. So we decided to evaluate our approaches on the mouse data generated by ENCODE, because in the current phase of ENCODE, the mouse data are quite well organized. So Bing Ren is the head of that mouse project. So there is a matrix. Let me just show you the matrix while I'm talking about it. So there is a matrix of embryonic developmental stages of mouse from embryonic day 10.5, 11.5, all the way to birth. And for each time point, the team has extracted out different tissue types to perform a large number of genomic assays, such as ChIP-seq of 8 histone modifications, RNA-seq, whole genome DNA methylation, so WGBS, and then for a third of the tissue time point combinations, DNase-seq. So it's a very nice matrix of data, and then the available data types are there, and then some of the boxes still says underway, but right now it's pretty much filled in. So there are 66 elements in this matrix, and with the whole set of data and then tissues being shared across the group, so it's actually really nice. In addition, so Len Panacchio's group and Axel Wiesel, so they have built this VISTA database of enhancers that are tested using transgenic embryonic mice. So as illustrated in this figure taken from Len's group, so you can take any predicted regions that might function as enhancers. You can take the region from human or from mouse, and then you make a DNA clone that contains that region connected to a minimal promoter with a reporter gene, and you take that DNA construct, you can inject into an oocyte of a mouse, and then you can implant that oocyte, and then to see when you have embryo mouse, to see when the reporter gene, where the reporter gene gets expressed. So you see this blue color from the reporter gene that will indicate tissue-specific function of the enhancer region you set out to test. So they have over 2,000 regions that have been tested, and over 200 regions each are being active in limb, brain subregions, and heart, respectively. So we basically took the data sets from his lab, from Len's lab, and used that to evaluate our very simple enhancer prediction method. So we can construct these true-false table, and you're very familiar with this, so I'm not going to dwell on this. And from that true-false table, we can make a precision recall curve, and the methods that score higher on this curve, area under the curve, would be a better method. So after several rounds of comparison, we came up with this very simple method that basically we're making genome-wide predictions. So we have to anchor our predictions on some epigenetic data. So we compared between the peaks of DNAs or the peaks of H3K27 acetyl. And we decided DNA's peaks are better choices than K27AC peaks. And how do we rank these peaks? So we decided that we compared a number of approaches, p-value, incorporating other histone mark signal, and so on. And at the end, we felt that the simplest way is to average the ranks of DNA's signal and the H3K27AC signal. So it's just a very simple, the most naive ensemble approach of two features. So as you can see from these graphical illustrations, so you have these DNA's peaks. They are actually quite sharp. Okay, so DNA signal indicates how sharp they are across the genomic region. And then you have H3K27AC signal. Remember, this is a histone modification. So the signal has to be on nucleosomes. And that's why you see two peaks indicating nucleosome positions that sandwich the DNA's peak, which indicates nucleosome-free region. Okay? So this actually indicates why the DNA's peak is a better choice for anchoring your prediction, because this is at the region that, in the middle of the region, rather than flanking the region. So what we do is we take these regions, and then we get different window sizes for DNAs and K27AC, with the K27AC window size being bigger. And then we can calculate the signal and then compute the rank. And then we can average the ranks, and at the end, and the regions that have the best average rank, we will predict all of them as possible enhancers. And in order to give a boundary, we actually take the K27AC peak boundary to assign to the enhancer. So at the end, we have enhancer-like regions such as this one, which has a peak location and then the width of it. So here is one example on a neural tube enhancer. And you can see this region was was part of our test set, so training and test set. And you can see the region that we can predict from the K27AC and the DNAs corresponds to this yellow region, agrees really well with a known VISTA enhancer that is active in neural tube. So after we developed this method, we met together with Len Pernacchio's group, and we selected 150 additional regions for him to do experiments. And the results have come out, so it is like a blind test, like the dream challenges here, because we didn't know the answer. He actually took the predictions and did the experiments, and the regions come out, a good majority of them are active. So that indicates the simple method is actually very predictive. So this is one of the example regions that we predicted it before he did the experiment, and you can see this region, this is exactly the region we predict. It is a limb enhancer that's active in limbs. So after that, so we have this method, so what do we do? So we decided as a consortium that we're going to go and build a registry of these kind of candidate regulatory elements. So we can predict enhancer-like regions, and likewise using a very simple approach for predicting promoter-like regions and insulator-like regions in hundreds of human and mouse cell and tissue types. And then we can compile all these regions together to make a registry, and we call it a registry of candidate regulatory elements. So we will also further make a tool for users to search, filter, and browse these candidate REs, candidate regulatory elements, based on other features, such as cell type, any kind of specific regular RE you are interested in, and then also identify the genes that these regulatory REs may regulate, so linked genes, okay? So here is an example for several tissue types. So GM12A7A is a lymphoblastoid cell type, and then liver tissue, T cells. So you can see that in these regions, the DNA signal shows very strong tissue specificity. And the peaks on the top are centered on DNA's master peaks. Master peaks means peaks across a whole compilation of cell types. So you can see these peaks. And then we combine that with K27AC signal to annotate possible enhancer-like elements in GM, but not in liver. As you can see, in liver, there's no signal, okay? Neither DNase nor K27AC. And we can do exactly the same across the board for enhancer-like regions, for promoter-like regions, but instead of using K27AC for enhancer mark, we now use K4ME3 for promoter mark. And then we can do that for insulator-like regions using CTCF ChIP-seq signal for insulator mark. So at the end, we compile everything into these registry I mentioned earlier. So there is a visualization method that we're building for people to browse and then download and filter these regions. And here is a link of the visualization tool. If you are interested in this, please give it a try. So you go in and then you can search them according to the cell types I mentioned earlier. And then there's a shopping cart feature. You can add whatever regions you have into your shopping cart, and then you can visualize them for details, for the expression profiles of genes within the same TED domain, or predicted to be linked with the RE, and you can compare different cell types, whether or not their regular candidate REs overlap with one another. So the key is for those folks who are very interested in transcriptional regulatory elements, these resources will provide tissue cell type information, and then will provide the activities of these candidate REs, and also intersect with the hundreds of TF transcription factor ChIP-seq data sets in the same cell type that ENCODE has produced, and then we'll link these candidate REs with possible target genes. So that is something we're working on, very busy, trying to wrap up the phase 3 of ENCODE. And just to give a little bit illustration as to what you can do with such a registry of candidate REs, I'm giving you a small example on annotating non-coding genetic variants associated with human disease. So there is a huge GWAS community out there identifying genetic variants of which majority are in non-coding regions. So how to interpret the possible functions of these genetic variants has been a major goal of the biomedical community. So we think that the ENCODE data could help with that endeavor. So in this example, there is a SNP in this particular one, which is in linkage disequilibrium with SNPs associated with multiple sclerosis. So this SNP is indicated here, and you can see that it is squarely inside a DNase peak, very nice DNase peak, and also inside an enhancer-like candidate regulatory elements in our registry. It is in an intron of this ZMZ1 gene, okay? And it was shown that in GM1287A, which is this lymphoblastoid cell type, these lymphoblastoid cell type DNase hypersensitive sites have been shown to be enriched for multiple sclerosis variants as a whole set. So this is a relevant cell type to be trying to interpret these multiple sclerosis variants. Furthermore, if you look at this variant very carefully, it actually overlaps with the binding site of a transcription factor called ELF1. So you can see that the variant here, the major allele is T, which agrees with the motif, and the minor allele, which might be implicated in multiple sclerosis, is C, that is not the consensus of the motif and might break the binding of this transcription factor. So, okay, that's all good. The candidate RE might be able to tell you the cell type, might be able to tell you which transcription factor binds there and whether or not the minor allele is going to break the binding of the transcription factor. But what next, right? So what might be the target gene of this candidate RE? So there are a number of methods for detecting target genes of candidate REs and many machine learning algorithms maybe some of you are working on. One really simple method is actually just correlation, and still many people still use it. So let me just explain very briefly. So you have this signal for the candidate-like, enhancer-like candidate RE region, okay? And then you have average signal, maybe histone mark signal, maybe DNA signal, maybe RNA-seq signal, whatever signal you may have in the promoter or the body of possible genes in the neighborhood. So what you can do is you can correlate these signals across cell and tissue types. So that's particularly pertinent for ENCODE because we have hundreds of cell types. So if you have this signal across a whole vector of 100 cell types, and then you have the gene information across 100 cell types, you can just correlate the 100 vector versus 100 vector, thinking that if the gene and the regulatory element, they go on and off in a synchronized manner, that's a strong support that the regulatory element may be targeting that gene. Okay, a lot of people do that. So we did implement that method. And also, in addition to that, we also implemented a random forests-based model for supervised machine learning. Why can we do that? So I didn't stress earlier that in ENCODE, we also produce this kind of chromatin structure data. For example, chia PET is one technique that predicts which regions of the genome tend to go together with other regions of the genome. But you can pull those interactions out using an antibody targeting RNA polymerase II in this example. Or you can target CTCF. You can target a number of things. And then also there is an ENCODE group, Job Decker's group, who has invented the high C, 5C, 3C type of techniques, also for chromatin structure. So we could collect all these data sets as the training set to train random forests algorithms. And so here are this is just a nice graph to show these 3D interaction maps, okay? So actually, the problem is actually quite complicated because these data sets actually differ quite a bit from one another. So even for very simple statistical properties, such as the distance between candidate RE and gene, if you look at each of the data sets in my previous table and make a histogram of the links, regulatory element and gene links, you can see that the different data sets differ quite a bit in how long the tail is for this distribution. And that actually affects the training quite a bit. Okay, so we have to have some kind of distance cutoff. Otherwise, the performance of your method will be quite affected by the long tail. So long story short, we came up with ways of doing the distance cutoff to construct positive sets of links, negative set of links, and then cross-validation, and then tested a few methods. So here is the baseline. So this is the baseline of correlation methods I mentioned earlier. So the elements and gene, if their signal are coordinated across a whole panel of cell types, you might predict they are linked with one another. So just the correlation method performs quite poorly. As you can see, this is the ROC curve of 0.68, and the PR curve has an area under the curve of 0.18, okay, if you just use the correlation, using the chromatin structure data sets as the gold standard. So we implemented two types of random forests models, depends on which features we put into the model. So one is a minimal model, which we put in features that are available for most of ENCODE cell types. And for the more comprehensive model, we additionally add additional epigenetic data sets that are available for a small number of ENCODE cell types. So here, let me just focus on the minimal model. So using the minimal model, you actually get a much better performance than just correlation. And here indicated by area under the ROC, now it's a boost from 0.68 to 0.92. And even more encouragingly, the PR curve now looks a lot more respectable. The random forest model also can tell you which features are important. Here is the rank of features by their importance. So the most important feature is actually the distance between the candidate RE and the gene that you might link to. In addition to that, so the K27 acetyl signal at the transcription start site of the gene is really important for predicting whether or not there's a link, which kind of makes sense because if you think your candidate RE is regulating a gene, that gene should be active, right? So if the gene is active, it would have a strong signal of K27 acetyl. Similarly, DNAs, and then there is, I can't quite see from here. So this is a correlation between, I think, K27AC. And so we basically putting the signal in the promoter, the signal in the enhancer-like regions, the correlation between them, and then the conservation. So bunch of K-mer correlation, which is a sequence feature. And you can see they all contribute to the model. And in total, the features, the signals at the transcription start sites are more important than enhancer features, which is understandable because the enhancer features have already been used once to predict these enhancers. So when we have this model, we can go back to the original example of these SNP for multiple sclerosis, and we can apply the model and see which genes did the random forest predict. And two genes are ranked the first and the second. The first one is actually, I mentioned this gene, ZMZ1. This is the gene for which the SNP is in the intron of this gene. And then PPIF is the next gene over, which hasn't been shown to be a target gene of this SNP. And if we do a PubMed search, you will find out that this PPIF encodes a mitochondrial permeability transition pore protein, which has quite a few support on, in animal models. It might cause autoimmune diseases, including multiple sclerosis. So this is likely to be also a target gene for the candidate RE that harbors this, SNP. So just to go back to the prediction, so you can see the genome in the genome browser. So the candidate RE with the SNP is here, and the gene that it's in is here, and then this predicted target gene is next door over, and you can see that in addition to the prediction, we also have Pol2 chia pad links that links this candidate RE with the promoter of ZM1Z1 and then the promoter of PPIF. And I just want to note that these chia pad links were not included in neither our training nor the validation set for building the random forest models. So they constitute independent support for the prediction. So I feel like I need to summarize. So give you a summary. I just told you the effort of the ENCODE Consortium for data production, for integrating in the results, for building the encyclopedia. So the future direction for my group is to, in addition to link candidate REs with target genes, we want to analyze how our method performs on other benchmarking data sets and make the predictions for candidate REs across all ENCODE cell and tissue types, and then build all these into the into the human and the mouse encyclopedia. And the human encyclopedia is available already at the visualizer I mentioned earlier. And we're in the process of building the mouse encyclopedia. And we are also developing additional data types to incorporate in there. And we have been showing the visualizer to quite a few diverse user groups to see whether or not people have feedback, to see whether or not they're useful. So if you could try it out and let me know how you feel, that would be great. So just to acknowledge the people who did the work. So there are 3 people in my lab, particularly Joe Moore, Henry, and Michael. They did vast majority of the work I just told you. And it's a wonderful thing to be part of a lively ENCODE consortium where lots of data are being produced and there's a lot of interaction between participants with computational groups, data producers, and the data coordination center. And we are constantly looking for talented students and postdocs. If you're interested in doing this kind of work, please come and talk to me. And if you have any questions about what I said, I'm ready to take them.
Great talk. Also, thank you for the ENCODE effort. I have a question on the ROC curve. You use the TE link to the genes. What are the benchmark data set you used?
Oh, so there is a whole table of data sets I mentioned.
Oh,
so
the CHIAPAD? And
that's right, that's
okay.
right.
So
all of these are also in the same cell type as the prediction. So it's very important to match things up on the same cell type.
Okay. So you put them together as a whole benchmark, or you?
We actually have tested on each of these data sets. The conclusions are qualitatively the same. Okay, so the details differ because the data sets actually measure somewhat different truth of gene enhancer links.
So another question is when you link the variance with the, for example, gene expression, so you so-called a signal, right, in the enhancer region.
That's right.
So you have to use the cells from different tissue. So how do you balance the, for example, the tissue specificity and the number of samples you use? Because when you calculate the correlation, if 2 fields in the same cell line, you have 2 field data available for the correlation, right? So if you have too much, then you need to use diverse cell lines.
That's right.
The signal may different.
Okay, so that is a really good point. Okay, that's why we tested you could actually correlate a number of features, right? You could correlate DNAs with the DNAs, DNAs with the RNA-seq, and then, so that is a really good point, and we did quite a lot of testing, you know, how many cell types do you include? Obviously, you can't include too few, because then the correlation is not statistically significant. We don't have the problem of having too few cell types. We have hundreds of cell types. So that's not a problem, but then, you know, some of the cell types are more correlated with each other, like lymphoblastoid from multiple donors. They're obviously correlated. So you might want to cluster them. So there is some nuance about how to do the correlation. We did do testing, and then, you know, overall they are predictive, but not great.
Okay.
So if you want details, we can talk offline.
Yeah, thank you.
Thanks, great talk. I was wondering, in your results, it looks like distance plays is quite an important feature, but not quite so much biologically meaningful. So I was just wondering if you've tried regressing out the distance and seeing if your random forest, if it predicts for like distal enhancers, what would be like the most important features?
Well, distance is a feature, but still it can predict distal enhancers, even when distance is a feature in the model. You have to
but you don't want to use, say if you don't want to use distance, right? If you're just gonna
you do want to use distance as one of the features, because roughly half of the time depends on the gold standard. Like from 30 to 60% of the time, the nearest gene is actually the target gene.
Yeah, yeah. So,
Yeah,
but
you
if I
just
was
don't want to just use distance, then, you know, what's the point?
Yeah, but I guess I'm wondering, like, if you were to predict enhancers that were, say, like more than 100 kilobases away, what would be the actual most important features? What histone marks would be kind of most important?
Right. So, you know, if we don't use the distance, the other features still contribute significantly.
In the same proportions, or is it
That is a
slightly
good
different?
point, because we have to use the distance, so we didn't really care about the proportion without the distance. But I do see your point, that the proportions could change.
We'll take one more question, but Anshul, while we're doing that.
Yeah, why don't you hook up?
Set up.
Okay, I have a question about how you prepare the negative training set for the promoter-enhancer interactions, because the distance between the promoter and the enhancer is the most important feature. So does the negative training set have the same length distribution as the positive?
Correct. They have to have the same distribution. So we did match the distribution of the distances. And also, as you can imagine, some of the links may be ambiguous. And we kept them out of both the positive and negative sets.
Okay, and what are the size of the positive and the negative data sets? Do they have
So
the
the positive data sets will be the links in, say, CheerPad, okay? And the negative will be the pairs that are not in the CheerPad, but in the same distance distribution.
Okay, I see.
Thanks.